BioGad, simply more knowledge. A podcast not just for biogeeks, informative and interdisciplinary.
Greetings and welcome back. I'm glad you're tuning in again and want to know how things continue with the energy utilization of prokaryotes. If you want to know more, you've come to the right place. If this is your first time joining us, I recommend listening to last week's episode before tackling today's. This podcast covers various topics in biology. My name is Ulrike Randl-Gadora. I am a physical anthropologist and teach biology at a general secondary school. Today we continue with part 2 on the utilization of energy. Last week I talked about how prokaryotes manage to make energy usable for themselves. We already discussed glycolysis, ATP, the rise in acidity, and the development of a proton pump to remove excess hydrogen ions from the cell. Prokaryotes have always found a way when a problem arose. I have divided the stages of evolutionary progress in this energy utilization into 13 stages. We've already covered 4 of them. Let's continue with stage 5. The electron transport systems became more efficient over time. Eventually, more protons were transported out of the cell than were necessary to maintain the pH value. Now we come to stage 6. Protons are not inherently a disturbance or unnecessary, but can be useful for something else. So the prokaryotes ensured that the protons, which had actually been unnecessarily expelled from the cell, flowed back into the cell. This process works via the ATP pump. These protons essentially drove the ATP pump in reverse. Over the course of evolution, this proton pump developed into an enzyme that supports the formation of ATP, in other words, ATP synthesis, from ADP and a phosphate group. ADP is adenosine diphosphate. This form of energy production is called electron transport phosphorylation. During phosphorylation, a phosphate group is reversibly attached to a molecule. And this form of energy production without the use of free oxygen, O2, was now sufficiently perfected. But this led to a new problem. Although the energy supply was secured, competition for carbon now began, and carbon is absolutely necessary for building cell components. The glucose in the primeval ocean, that is, the grape sugar, C6H12O6, was running low, and that of course posed a problem for glycolysis, which could now only function in a limited way. What was needed? A new source of carbon. The primordial atmosphere contained enough carbon dioxide, CO2, but CO2 cannot be tapped so easily. It must first be reduced to extract carbon from it, and this requires suitable reducing agents. And these were becoming scarce. Not just any reducing agent can be used. That brings us to stage 7, reduction of CO2 from the primordial atmosphere to organic carbon compounds. So the primordial atmosphere contained enough carbon in the form of CO2. This carbon could be incorporated into organic compounds via reduction, but only as long as suitable reducing agents were available. Less glycolysis also meant less H2, that is, less molecular hydrogen. Of course, there were other reducing agents, and in sufficient quantities, but hydrogen sulphide, H2S, and water, H2O, are far too weak to reduce CO2. To summarize simply, it was possible to build a system for energy production and then modify it so that prokaryotes did not harm themselves, and that the system could function long-term and even yield surplus energy, provided that demands did not increase. And now comes the catch. If the number of individuals increases, larger amounts of building material are needed to form these cells. But since building material doesn't just lie around, it has to be synthesized from other materials, which requires suitable tools. So the prokaryotes were looking for tools that could fulfill this purpose. Stage 8 involves obtaining molecular hydrogen, H2. A new reaction had to be found because glycolysis alone was no longer sufficient. Several new pathways were discovered, 3 of which I would like to present. One strategy involved the use of radioactivity. This was confirmed in 2008 in a water sample from a gold mine in South Africa, 1,500 meters deep in the Earth's crust. Chemoautotrophic bacteria called Desulforudis or Daxviator were found. These bacteria reduce sulfates to sulphides, a process called desulfurication. The electrons required for this come from the radiolytic splitting of water. This process produces the reducing agent H2, i.e., molecular hydrogen, enabled by the presence of decaying uranium. A second strategy was to utilize rock transformation. When a mineral is converted into another mineral in the presence of water, the water is split and hydrogen is released. This type of mineral transformation occurs during so-called serpentinization, in which olivine is converted into serpentine. Even today, the hydrogen thus released is used by chemoautotrophic bacteria that generate energy from hydrogen sulphide, H2S. In the 1990s, traces of bacteria were found deep in the Earth's crust for the first time. These bacteria decomposed the rock. Bacteria living in this part of the Earth are referred to as endoterrestrials. Since this discovery, new chemoautotrophic variants of energy production are continuously being discovered. A third strategy is the use of deep-sea volcanoes. These are located several thousand meters deep along the edges of tectonic plates and are sources of CO2 and H2S. At these depths, it is pitch dark, water pressure is extremely high, and the water temperature is about 2 degrees Celsius. Submarine volcanoes supply minerals such as iron, zinc, copper, and nickel, as well as silicon in the form of quartz, SiO2, and sulphur in hydrogen sulphide, H2S. These volcanoes also release large quantities of essential building blocks for organic molecules from deep within the Earth. Due to the high temperatures of magma, these components dissolve in water at over 300 degrees Celsius and are discharged through chimney-like structures up to 10 meters high. When they emerge and meet the cold surrounding water, they precipitate. This precipitate looks like smoke, though it isn't. Depending on the chemical composition, it is either black or white. If you're interested in how and which substances are brought to the surface from seawater at these hydrothermal vents, I recommend a link you can find in the episode notes. Deep-sea volcanoes provide bacteria with vital reducing agents. Bacteria also produce DOM, dissolved organic matter. This is a dissolved substance found in soil and oceans. DOM stores enormous amounts of carbon, an estimated 660 billion tons, more than all terrestrial plants combined. Deep-sea volcanoes render this carbon bioavailable by breaking down DOM through high temperatures. Who were the first? Today, the bacteria that managed to meet their energy needs through the oxidation of H2S are considered the first chemoautotrophic organisms. This form of energy production is independent of UV light and is called chemosynthesis because the energy is derived not from light but from chemical molecules. Prokaryotes enable further development. As early as 1977, temperature-tolerant mussels and sessile tube worms were discovered at hydrothermal vents. These tube worms live in symbiosis with bacteria that settle in their tubes. One such species is Riftia pachyptala, a beardworm that grows up to 2 meters long. These worms host chemoautotrophic sulphur bacteria as endosymbionts in the trophosome of their body cavity and have no digestive tract in adulthood. Without these bacteria, they starve. They lack both a mouth and an anus. Hydrogen sulphide binds to hemoglobin in their bloodstream and is transported to the trophosome, where sulphur bacteria supply the worm with carbon. In Riftia pachyptala, this endosymbiosis was discovered for the first time. This species remains the model organism for symbioses with sulphur bacteria. Deep-sea volcanoes are considered the cradle of life. In the 2022/23 school year, I carried out a deep-sea project with my then Year 9 class at the grammar school. The project was developed in collaboration with Monica Bright, Austria's leading marine biologist. While Monica Bright conducted the project we had designed on the seabed of the Eastern Pacific, she and her research team also discovered a new ecosystem, the suburbs. This is an ecosystem located beneath or inside the seafloor. I'll talk more about this school project in the next episode. We now come to stage 9, the utilization of UV light. Again, prokaryotes came up with more than one solution. Strategy A, use of H2S. The ancestors of today's green sulphur bacteria developed a groundbreaking solution. They used hydrogen sulphide, H2S, via an indirect pathway. With the help of sunlight, they managed to transfer the proton from H2S to a molecule also present in large quantities, NADP. NADP stands for nicotinamide adenine dinucleotide phosphate. These prokaryotes thus built a new and significantly stronger reducing agent. The new reducing agent, NADP.H2, was strong enough to reduce CO2, that is, carbon dioxide. These prokaryotes were therefore the first photoautotrophic organisms. Where do these green sulphur bacteria live? If you want to find them, look for a water body containing H2S, hydrogen sulphide. You'll recognize it by its smell. It stinks like rotten eggs. In addition, such waters must contain little free oxygen and allow only limited UV light to penetrate. These bacteria cannot live in total darkness like chemoautotrophs, but thrive in low-light areas where other photoautotrophic organisms cannot survive. They utilize UV light thanks to special organelles, chlorosomes, and special pigments, the bacteriochlorophylls. With this support, UV light can be used extremely efficiently. That's why green sulphur bacteria can carry out photosynthesis in such environments. However, they do not have chloroplasts. Those are only found in plants. Strategy B, use of H2O. A similar strategy was employed by the Cyanobacteria blue-green bacteria, but instead of hydrogen sulphide, they used H2O water. When water is split, O2 oxygen is released. The advantage of H2O is obvious. It was practically unlimited and available throughout the oceans, not just near deep-sea volcanoes. However, there's a catch. H2O has a much more positive redox potential than H2S. The difference in redox potential between H2O and NADPH2 is even greater than that between H2S and NADPH2. This energy gap is difficult to overcome. The photosystem developed by the ancestors of green sulphur bacteria simply wasn't sufficient. Stage 10. The Cyanobacteria found a solution. They connected two photosystems in series. Something similar is still found today in higher plants. This form of photosynthesis was so successful that free oxygen, O2, began to accumulate in the atmosphere. Oxygen levels rose to 2%. That might not sound like much. Today we have about 21%, but for that time it was quite a lot. This oxygen also caused the previously dissolved and reduced iron Fe2+ in seawater to oxidize. It precipitated as red Fe3+ and settled on the black basalt of the seafloor. In other words, the iron began to rust. Over time, massive layers of banded iron formations were created. This great oxidation event occurred about 2.4 billion years ago. But now a new problem arose. Too much O2 is toxic and led to the first global environmental poisoning and mass extinction. O2 is a strong oxidizing agent for all organic compounds and can even cause cells to die. The amount of oxygen a cell can absorb through its membrane depends on its surface to volume ratio. Don't worry, no maths example today. Simply put, the smaller the cell, the more oxygen it can absorb. This may have contributed to the evolution of multicellularity. Stage 11. To neutralize oxygen, it could have just been detoxified and left unused. But the prokaryotes did better. They not only detoxified oxygen, but managed to use it. They developed a mechanism as atmospheric O2 levels rose to about 1% of today's levels. These prokaryotes used oxygen to break down organic compounds in the cell. They oxidized substances and simultaneously gained energy. How? They used the transport systems from stage 4, remember, the ones involving ATP. These systems were modified to enable the transfer of electrons from NADLH2 to O2. There are still bacteria today operating at this evolutionary level, the purple bacteria. Stage 12, gaining energy using atmospheric oxygen, was such a major breakthrough that, about 1.5 billion years ago, anaerobic prokaryotes, those that do not require free oxygen, internalize the strategies of aerobic purple bacteria. They simply engulf them. The purple bacteria became endosymbionts. Over time, these internalized bacteria evolved and transformed into the powerhouses of the cells, mitochondria. There is also another fascinating hypothesis about the origin of mitochondria. According to this, Asgard archaea were the first to contact these symbionts, then surrounded and engulfed them. Finally, they enslaved the symbionts and captured their genes. This, of course, is quite an anthropocentric way of putting it. By this time, prokaryotes had already established glycolysis, photosynthesis, and oxidative metabolism. Now, endosymbiosis paved the way for the emergence of eukaryotic cells. Incidentally, the amoeba species Pelomyxa palustris has no mitochondria but lives in symbiosis with aerobic bacteria. Stage 13. Photosynthesizing cyanobacteria later followed a similar path as the purple bacteria. They entered eukaryotic cells and became chloroplasts. Many eukaryotic cells still contain cyanobacteria. Modern prokaryotes show an astonishing diversity of metabolic pathways, far exceeding those of all other organisms combined. For 2 billion years, they were the only living beings on Earth. The ancestors of eukaryotes were probably anaerobic loci-archaea, which belong to the Asgard archaea supergroup. These archaea are named after various Norse gods, including Loki, Thor, Odin, and Heimdall. Loci-archaea were discovered in 2010 at a black smoker in the Arctic Ocean, known as Loki's Castle, hence the name. Prometheoarchaeum syntrophicum is a species of Loci-archaea that proves archaea can form complex syntrophic relationships, meaning this archaeon lives in association with other organisms and the partners exchange metabolic products. Eukaryotes probably arose around 2 billion years ago through symbiogenesis, the merging of two or more archaea. If we look more closely at evolutionary processes since the emergence of eukaryotic cells, it appears that evolution has primarily focused on adapting to the following conditions: increasing atmospheric O2, changes in the Earth's surface due to plate tectonics, and shifts in climate. Added to this is the adaptation to ever-increasing ecological differentiation of habitats, which, thanks to humans, is now once again declining. What conclusion can you take away today? Nature always finds a way to solve a problem and pursues various strategies to do so. The interior of our planet, together with water, is therefore the source of life. Whether life first arose in the sea or deep in the Earth's crust is still unclear. There are countless hypotheses attempting to explain Earth's geological conditions. One, for example, asks why continents exist at all. The transformation of heavy basalt into lighter granite may not have occurred purely geologically, but perhaps also with the help of bacteria. Bacteria may even play a role in the genesis of earthquakes. Many of these scientific ideas are the reason we search for life on other planets, and why water is always a focal point. The emergence of eukaryotes was only possible because the continents also emerged. Their erosion brought phosphorus, essential for genetic material, into the oceans. If you ever hear anyone moaning about why they have to learn all this chemistry for biology, from today onwards, you'll have some arguments to counter that. I hope you were able to take away something useful today as well. A few additional links can be found in the episode notes. Catch you later.
If you enjoyed this episode, please give me a thumbs up. If you have any constructive feedback for me, feel free to send it to bioget@outlook.de. Thank you.
This was an episode of Bioget by Ulrike Randl-Gadora, covering various topics in biology.
The text was originally written in German and then translated and narrated by AI. If you notice any translation errors, please send an email. Thank you.